Myself Vikas Gowda. Warm welcome to our YouTube channel StudyWire. Today let's discuss about cell ultrastructure. You people know what is cell. It is the structural and functional unit of living organisms. You know the diameter of a cell is 0.2 to 0.5 micrometer. The shape is varies from cylindrical, spherical, rod-shaped and hexagonal, irregular shape. And bacterial cell size is 0.2 to 0.5, same as normal cell. Next, come to there are two types of cells, you know, animal cell and plant cell. In what is the difference of animal cell and plant cell? In animal cell, cell wall is absent. In plant cell wall is present. And in animal cell, chlorophyll is absent. In plant cell, chlorophyll is present. This is the characteristics difference of these types of cell. In animal cell, vacuoli are small and multiple. In plant cell, vacuole are large and single. In animal cell, centriole is present. Here centriole is absent. Here lysosome is present. Here lysosome is absent. Here lysosome is present. And here centrosome is absent. Here centrosome is present. It is also a characteristics difference of these types of cell. And here scleroplasma is absent. Here scleroplasma is present. Here plasmodesmata is present, and here plasmodesmata is absent. Now let's move to next part of our cell. Let's discuss. Here we are dividing lecture into many parts. Let's discuss about cell wall in plant cell. Okay. Generally, you know, cell wall is the first layer of cell. Here cell wall is present only in plant cell. Here in cell wall, three layers are there. First one is middle lamella, next primary cell wall, next secondary cell wall, then plasmolemma or plasmomembrane. Okay, let's study each part. First one is middle lamella. In middle lamella, pectin is the main protein. Pectin is the form of Mg2 plus and calcium 2 plus salts. Pectin is hydrolyzed by strong acid and pectose. As you know, cellulose is absent in middle lamella. This is the main characteristics of middle lamella of plant cell. The first part to originate cell division. It is the main point to your exam points. It is the first part to originate in cell division. Okay, that is primary cell wall. It is constituted of hemicellulose, cellulose, pectin, protein and lipid. Hemicellulose of 30%, cellulose of 30%, pectin of 5% and protein of 5% and lipid of 7%. It is deposited after the middle lamella. You know, first one is middle lamella, second one is primary cell wall, next layer is secondary cell wall, next layer is plasmolemma. It is deposited after middle lamella. The cells in meristematic tissue contains only primary cell. This is also important point for your examination. This may ask in MCQ. The cells in meristematic only constitutes only by primary cell wall. Okay. Next, primary cell wall is not deposited near plasmodesmata. This is also an important point. It is not deposited near the plasmodesmata. You know plasmodesmata? Plasmodesmata means it is the way where the nutrients are passed from cell to cell. Now, let's go to next part. Secondary cell wall. It is composed of cellulose. Positions in the layers are very important for examinations. Now, in secondary cell wall, it is composed of cellulose. In many tissues, lignin and suberin are present in many tissues. In few tissues, cutin is present. Cellular microfibrils are packed and arranged in the parallel manner. This is also a point for examinations. They may ask cellular microfibril packed and arranged in perpendicular manner in cell membranes or cell wall. These statements are true or false. Like statement based questions they may ask. This is very important. Cellular microfibrils are arranged in parallel manner. Next layer, plasma lemma or plasma membrane. It is composed of lipids and proteins. As I told, every compositions are very important for exam point of view. Okay. In lipids, there are many types are there. One is phospholipid. It is in phospholipids there are three types. One is lecithin. Next one is cephalin. Another one is spigomycin. Next, for after lipids, many lipids are constructed. What are the lipids means? Cholesterol, ganglioside. Other one is triglycerides are also present and common fats are present in lipids. These are all composition of plasma lemma. You know plasma lemma or plasma membrane which is located after secondary cell wall. First one is, as I mentioned, first one is middle lamella, second one is primary cell wall, next one is secondary cell wall, next one is plasma membrane. Okay, these are all the arrangement of cell wall. Many scientists give many view of their point, that point of view. For example, Robertson gives unit membrane for arrangement of cell. This is a concept, their views. Danson and Davell gives lipid bilayer and phospholipid bilayer. And finally, Singer and Nicholson gives fluid mosaic model. This is the universally accepted model. Universally accepted model. So this is important for our examinations. Okay. Next, go to another concept, that is unit membrane. Let's elaborate. As you know, this is derived from Robertson. There are three distinct visible layers. What he told means, in cell wall there are three distinct layers are visible. One is dense and osmophilic. Next one is osmophilic light of 20. This one is 20 Armstrong, you know, length. And another one is same as dense osmophilic. Last layer is also a dense osmophilic of 20 Armstrong. In middle layer, this is osmophilic. This one is light. These both are thick wall. This is rejected because he didn't explain deeply, and simply they remember the name, that is Robertson. It is given by Robertson. He told the cell wall is composed of three layers. First one is dense and osmophilic. Second layer is osmophilic, that is light of 35 Armstrong. Last layer is dense and osmophilic of 20 Armstrong. that after rejection of unit membrane, lipid bilayer, phospholipid is came to existence, that is lipid protein bilayer or lipid protein bilayer. Or lipid protein bilayer. It is came in effect of 1935 by Dalton and Dawel. As you people know, this is lipid bilayer. External one is hydrophobic end. This is hydrophobic. This is hydrophilic. Means water loving. It is just nothing but arrangement of cell wall. It is arranged by lipid bilayer. He explained Dansel and Dravel. First one is unit membrane, which is explained by Robertson. Cell membrane is made up of lipid bilayer. As you know, this theory also rejected. Then come to, as you know, phospholipid bilayer is bilayer. First one is protein, next one is lipid. Protein, lipid bilayer. In middle there are in middle protein is present. In middle protein is present. This one is hydrophilic end, which means it loves the water. Hydro means water, philic means love. Hydrophilic head. This one is hydrophobic tail. Part is fluid mosaic model. This is proposed by Singer and Nicholson. This is the universally accepted model, as you people know. Here, in previous one, he only taught us what the cell membrane is made up of. It is only made up of hydrophilic end and hydrophobic ends, lipid and protein bilayer. But here some other concepts are also there. He didn't give about protein. What are the proteins? What are the layers? He didn't give. That's why that model also rejected. Finally, lead mosaic model is came to effect and universally accepted model. There are two layers with lipid and protein molecule. As you people know, this is hydrophilic end, hydrophobic tail. These are all protein molecules. What you people see, these are all protein molecules. And it is compactly arranged. This is bilayer. They are compactly arranged because it gives structural and functional support to cell. Therefore, they are compactly arranged and rigidly arranged. Many protein complexes are there. For example, carbohydrates. Glycoprotein is one of the main protein of fluid mosaic model, where we seen in cell membrane. Here, protein acts as receptors. Receptors means it receives the signals from the environment. Here, protein acts as receptor. 1 minute. Your protein acts as a receptor. Means it receives the signals from the external environment to the bilayer, and this is composed of this fluid mosaic model is widely accepted because he cleared all concepts which are doubted by everyone. Therefore, we considered as universally accepted model.